Good morning, everyone. Thank you so much for joining us for our webinar series. So for this month, we are very lucky to have such a distinguished speaker who are real expert in single cell to join us today. So let me briefly introduce the first speakers, Associate Professor Ponpan Matangkasombut Choopong. She's actually our alumni. She's got her MD from Faculty of Medicine, Chulalongkorn University. So we're very proud of her. And she, have got a long list of CV. She went on to get her residency training. So she get both her American Board of Internal Medicine and Fellowship of American Board of Allergy and Clinical Immunology from Metro West Medical Center and National Jewish Medical and Research Center from the US. And then she didn't stop. She's pursuing her basic science career by going for a Ph.D. So she got her Doctor of Science from Harvard School of Public Health and continued to do her postdoc at Children's Hospital Boston, Harvard Medical School. And just recently, she's go back to do her research at Wellcome Sanger Institute at the UK. And I think this is why she's going to talk to us today about her role in the single cell world. Her research are focusing on infectious disease, particularly dengue infections and other immune-mediated disease, and recently in COVID-19 as well. So thank you so much, Ning, for joining us today. And I would like to also introduce our young scientist, but his CV is, extraordinary as well. So Dr. Waradon Sungnak, I forgot both of them, come from Department of Microbiology, Faculty of Science, Mahidol University. So Dr. Waradon got his bachelor degree in biochemistry from Columbia University and also got his Ph.D. in immunology from Harvard University. And he's doing his postdoctoral training at the Wellcome Sanger Institute. And Dr. Waradon, despite very young age, and he just joined us in Thailand, right? But he has several prominent papers in Nature Medicine. And I think we're going to see a new paper in Nature as well, right? In single cells on dengue and the COVID-19. And today, despite their busy schedule, we are very happy and very thankful for both of them to join us and will tell us more about the role of single cell. So their title is Immunology in Single Cell Genomic Era. So thank you so much, and the floor is yours.
Thank you, Pen, so much for inviting us here today. And we're very proud to present you our single cell work, and hopefully we can provide some basics on single cell in immunology. So that's the title of our talk, and thanks so much for your kind introduction. So we'll talk on the immunology in the single cell era. So the outline of our talk, I'll start first by the introduction of single cell omics in immunology as an overview. And then Ajahn Waradon will dive into more detail in the application of single cell omics analysis and frontiers in single cell omics. And then I'll come back and tell you a bit more about Human Cell Atlas project and Immune Cell Atlas impact in human health and disease and our own experience in Thailand. So first of all, we're all immunologists, and we all know the immune cells has special characteristics like its diversity, specificity, plasticity, and adaptability. So immune cells are very heterogeneous. So single cell technologies include assessment of both transcriptomes, genomes, proteomes, epigenomes level of individual cells. So it's ideally suited for studying these properties of immune cells. So why do we use single cell omics in immunology? So we use it to dissect heterogeneity of immune cells. So compared to bulk RNA sequencing, where you pull every cells in the tissue and put in the tube and extract RNA of all the cells together. When you get RNA-seq results, you get an average gene expression from all the cells. So it lost the cellular heterogeneity. In other words, when we use single cell analysis, we will separate each cell in a single cell input. So we'll get RNA-seq of each individual cells. So we are able to dissect the heterogeneities and different in transcriptomic profile of each cells. So before the advent of the single cell RNA-seq, we usually use FACS. That's also single cell, but we have limited number of markers, protein markers of 17 to 40 cells. And then afterward, we have CyTOF that can assess maybe 40 cells. Oh, sorry. Let me get that. Okay. But at any rate, this single cell RNA-seq, FACS, and CyTOF are complementary so that we can have transcriptomes and proteins markers at the same time. But at any rate, we also have this newer technologies called multi-omics or site-seq, app-seq, which can also assess proteins level, which Ajahn Waradon will tell you a bit more detail in a little bit. So if we look at single cell RNA-seq protocol, we'll separate cells into individual cells with various techniques such as droplet-based, microwell-based, nanowell-based, or just FACS sort into plates. So after we separate individual cells, a special technique to amplify this minute amount of RNA and tag them with cell markers, and then we amplify and we sequence. So the first one that I would introduce you is SMART-seq2 protocol, which we simply use FACS sorter to sort individual cells into each well of 96 well or 384 well plates. And then we index each well by like an extra indexing so that we can track which RNA is from which cells before library preparation and sequencing. So in a more commonly used one is the droplet-based technique that we utilize droplet-based. So each cells will be separate by this droplet of water in oil emulsion, and each cells will be tagged, separate with one bead, and this bead contains barcode and also poly-DT that will bind to poly-A of each mRNA. So we can track each cells, each mRNA from which cells from the DNA barcode. So after cell separation into beads, it will undergo PCR, cDNA and PCR, and also library preparation. And then with the analysis pipeline, we can track back which RNA is from which cells. So this is just to compare various protocols, such as this SMART-seq2, Fluidigm C1, and the droplet-based. So in the SMART-seq2, it's good in a way that you can FACS sort it, the cells that you're interested. You have a high depth, but it costs a lot per cells. But a microfluidic base, you can sequence like up to 5,000 to 10,000 cells at a time. And the cost per cells is actually lower. And a lot of us are afraid of the cost, but newer approach drive the cost down, and we are now able to do multiplexing. So each reaction, we can put several samples in. That drive the cost down quite a lot. So overall, what can the single-cell omics data tell us? Ajahn Bloom will tell you about this in much more detail, but just to capture a big picture, it can help us identify new type or new cell states, look at cellular network and cell-cell interaction, look at tissue adaptation, localization, evolution, compare between individuals, disease versus health, severity of disease, for example. It can also assess the molecular pathways of immune response, predict trajectory, and also can look at T cell receptor and B cell receptor diversity and clonality. Okay, now I'll turn the floor to Ajahn Waradon.
So now that we seen some basic ideas about why single-cell RNA sequencing is giving us a certain aspect that traditional bulk RNA sequencing could not provide us based on what Ajahn Ponpan mentioned, then we delve deeper into the different aspect of capabilities that single-cell RNA sequencing would provide in multiple analysis aspect. So before we get into that multiple capabilities, and I'd like to show you all a little bit about what we should expect in terms of the result from single-cell RNA sequencing versus the bulk traditional RNA sequencing. So in the traditional bulk RNA sequencing, so as Ajahn Ponpan mentioned, right, we will get gene expression profile of aggregates of cells in a given sample. So what we will get in the end would be the gene expression profile of each of the individual samples. So the result would be something like, what I'm showing here, which is the gene expression table, where here a row would be a gene, and then a column would be a sample, and then the number that you get for a given cell would be expression of each gene per that sample. So that's the aggregate. And with this kind of result that you get, what you could do next would be to compare which genes are highly expressed in sample 1 versus sample 2, and in what pathway are being represented there, right? But here it's going to be based on the aggregates of all cells within that particular sample. But if you're doing single-cell RNA sequencing, what we are going to get instead would be the gene expression profile of single cells, so individual cell within a given sample. So now I hope that you can visualize what kind of thing we could do more, right? So extra step that we are capable to do now is first, right, to be able to characterize what cells we have in each of the samples. And once we characterize the cells that are in there, then we could take a look at the gene expression profile of each of the cell type. And next, if we're going to compare differences across sample, then we can ask, like the comparison between the yellow cell from sample 1 versus the yellow cell from sample 2. So now we could see that with this higher resolution, the comparison would be a lot more appropriate, because now we're going to make a comparison with the matching cell type instead of doing the comparison across heterogeneous samples that we have now. So now we could see that, like it's not going to be useful in terms of cellular phenotyping that would infer the cell identity within each other sample. But then with the comparison, let's say from the healthy versus the disease samples, then the comparison would be fairer, because now we would be able to make a comparison in the cell type specific fashion. So this is a standard common analysis pipeline. Well, because once we get the sequencing results, then we get the reads that would be mapped into a reference genome and transcriptome, and then we would quantify the expression of gene per individual cell. Once we get that, we do some quality control, making sure that we would only select the profiles from cells with high quality. And then we do some normalization to make sure that comparison across different cells would be fairer. And then, as I mentioned, right, like the next thing that we would be able to do is to identify what cells are in there. And a common approach would be based on something called dimensional reduction and clustering, right? We have different cell type, and the same cell type would be clustered together based on a similarity of gene expression. We can do other things. For example, instead of identifying discrete cell type, we can model differentiation in the sample that we have would contain cell that have phenotype that are more like a continuum, for example, in differentiating cell, or we can do some other things, so things that I would like to talk about in a bit. For example, inferring cell-cell communication, inferring genes regulatory network. So just to recap, right? So what single-cell transcriptomic would provide us more is not just to provide us in terms of the changes in differential gene expression. So now we can do what's called cell atlasing, which is to identify cell types or cell state in a given environment in a much less biased manner, right? So instead of relying on a few markers, now we just use transcriptomes, right? The entire gene expression to infer the cell phenotype or the cell behaviors. And once we can get the identification and notation of the cells, then we can do systemic phenotyping of cell across different conditions in a cell type specific manner. So now we could pick, okay, this is macrophage from sample 1 versus macrophage from sample 2, and then we could do macrophage comparison instead of doing comparison in a bulk fashion where you might have confounding effects from contamination of cell types that would make a comparison less appropriate. So to step back a little bit and think more in an abstract fashion, right? So what is actually a cellular identity, right? So there's a very nice paper arguing in terms of a more abstract fashion that when we think about an identity of a cell, we can think of it as integration of multiple identity vectors. And the vectors can be attributed to multiple aspects of cellular phenotype and identity. For example, it can be based on the discrete cell type, right? Whether it's a T cell, macrophage, B cell, or it can be attributed to continuous phenotype. So let's say, you have a differentiating T cell, so it's naive, it's transitioning, it's a memory cell, it's an effector cell. Or you can have cell identity based on unidirectional temporal progression based on differentiation, for example, from the stage ranging from hematopoietic stem cell differentiating into multiple lineages, T cell, lymphocyte, neutrophils, or erythrocytes. It can be based on where the cell are in the cell cycle, whether it's resting cell or it's proliferating. Or it can be based on where they are in the spatial context. Like when the same cell type is in location 1, it has a given phenotype, but then when it moves to another location, then it can acquire a different phenotype based on where they are. And this is not just due to the cell itself. It can be reflected through their changes upon environmental stimuli. So once, for example, so this is obvious in the context of immunology, right, where we have an immune cell before and after infection. So the phenotype could change. So you could see this, right, that, like there's so many aspects of cellular identity. And the idea is that transcriptomes would be one of the major sources of changes or differences that we can capture any of this, right? So this is why a transcriptome could be very, very powerful, because we can capture a lot of information and we can model based on this different aspect that we can infer things about their biological implications. So this is an example of, like the discrete phenotype, right? So this is based on, this is actually our data from PBMC, and this is just based on the clustering, right? So based on the transcriptome, these are this discrete cell type that we can identify, and then you can see them in different clusters, right? So cells that are similar in terms of their transcriptomic profile, they would, like cluster together, and then we can label, right, each of the different cell type here, and you can see the resolution that we can get from the single cell RNA sequencing data. And this is why, right? So when we take a look at single cell RNA sequencing data, we see dots with color. And one of the most popular analysis tool is known as Seurat. And Seurat is actually a French artist, like during the kind of impressionist era. And the reason why it's called Seurat is because like this particular artist has a style called pointillism, where you can see that he used dots, right, in colors, and dots in color represent his subject matters, right? And that's similar, right? That's a comparison or analogy to single-cell RNA sequencing, where we typically represent our data in color dots to convey the biological messages. So that's just a side note. So now, once we get the clustering, right, and try to annotate cell type, and now we try to make maps of different cells in our given sample, then we can identify the cell types. So in the past, right, like where we just generated the data, and then we try to identify cell type based on the information we have from our sample. That's what's called unsupervised cell type annotation. So just identify cell types of cell states based on cluster within a given experiment. However, now the field is moving more toward more of a supervised fashion, where once we get more and more data, and we get more and more accurate annotation of cells, then instead of just like every single time when we do new analysis and we try to do cell type annotation by our hands within our own sample, we use what people previously published and use that to kind of train a model, and then use that model to infer cell type annotation in our sample. So the field moves more toward a supervised cell type annotation, where based on the highly accurate, good supervised reference, we use that to infer the cell type there. So there's a tool, for example, Azimuth, where there are some highly accurately curated references based on like some of the multi-omic data sets. And now for certain samples that we have, so if they are kind of equivalent, for example, if we have PBMC single-cell RNA sequencing, so instead of having to annotate the cells ourselves, we can use their highly curated reference and then inferring the cell type identity of our own data sets based on their reference. So this is known as reference-based cell type annotation. So the reason I'm talking about this and bringing this to the audience here today is to just show you that while the field is moving, the way that we analyze the data is also changing. So this is a kind of like a Bayesian way of thinking, where when we get more and more data, the capability of us being able to analyze or doing the analysis is getting more and more powerful. And that's emphasized the reason why we should be more and more active in terms of contributing to the references themselves. And that's a rationale for why we have been active in trying to push for generating references based on data from Thai people's populations. So I'm providing you more of the examples on like what things can be done in terms of cell type identity. So showing you Atlas. So this is early fetal maternal interfaces from one of the published paper showing cell type identities in the placenta decidua based on the fetal tissues. And there's also a recently published paper which is called Tabular Sapiens, where they collected samples from a given individual, but across different tissues, and with the expectation of trying to do the mapping of multiple cell types from multiple tissue in a more controlled fashion from control individuals. So in the end, it got 500,000 cells from like multiple tissue from the same donor. So with this, right, with the control of author confounding factors, this is expected that the cell type annotation would be more accurate. So things are not just hold true for the fact of atlasing of the healthy tissue. The atlasing can be done in the disease context as well. So this is the cancer immunotherapy. So I'm showing you some results or some literature related to cancer, right? So this is an example of an atlas of cancer-associated T cell, where scientists can harvest cancer from multiple types, and then just explore the T cell from these multiple cancer types, looking at gene expression, looking at the TCR sequences, and then kind of infer any biological phenotype from the T cell there, exhaustion phenotype, effector phenotype, and then inferring the similarity and the differences across different cancer tissues. So besides a discrete cell type annotation that I just mentioned earlier on, I also mentioned that sometimes cell type identity is not just based on discrete cell type, but if your sample have differentiating cells, right, like there would be a phenotype of cell that are more like a continuum, and we can actually model that. So if like sample that you have differentiating cell, so then things would be more like a continuum rather than a discrete cluster. And with that, you can infer what's called trajectory, right? Like where can you mark the beginning, right? And how cells just accumulate differences until the terminal differentiation state. And then this is how, right, you can model how cell differentiate, how many lineages are there, and then what would be the differentiation paths that you can infer from the sample. And these are some of the examples. So this is a paper, a preprint published like last week, where they model like human neural organoids. And you can see that, like with the sample there, they can model things and can infer a kind of differentiation trajectory based on the sample that they have. So beyond that, we can also model cell-cell communication. So what I mean by cell-cell communication here is that when we get a gene expression profile of a transcriptome, we also knew based on our prior knowledge on the receptor ligands, right? And with that kind of information, we've known, for example, like this IL-10 and IL-10 receptor, and then we can model which cell express IL-10, which cell express IL-10 receptor, and we could model some kind of correlation and infer whether there are cell-cell communication in there. So there are multiple tools that can address this kind of research question, cell-SHAD, cell4DB, and then I'm showing you, like an example that, like we can model receptor ligand expression cell that make a ligand, and then which could be the recipients of the ligand based on the expression of the receptor in a correlated fashion. We can integrate the single-cell RNA sequencing data with genetic differences. So right now, people, as Ponpan mentioned, that we tend to multiplex sample, right? Pull multiple sample into a single 10x length. So typically, when we pull a sample from different donor, we also use SNPs to kind of demultiplex cell from multiple sample later on. But then when we have SNP information, we would be able to infer genetic differences as well, right? So with this kind of information, right, we can link between the SNP variability and the immune variation at cellular level, right? So we can infer things like, like this particular SNP variance is related to differences in expression of gene X in cell type Y, for example, right? So this could result in us being able to infer genetic driver of immune cell phenotype, and this has become a more and more powerful tool for us to understand a causal relationship between genetic driver versus the cellular phenotype, and then linking to things like diseases. For example, in this particular paper, they're showing potential causative genetic factors that could contribute to autoimmune disease. So now, in terms of the application, we mentioned a little bit about using single-cell RNA-seq to do some kind of atlasing, but we can also use this to apply in a more, like disease context as well. So I'm just going to give you some examples based on ours and other people's work. So as I mentioned, right, like an additional thing that we could do besides cell atlasing is to do systemic phenotyping, looking at changes or differences in a gene expression profile in a given cell type across multiple samples. For example, looking at differences in cell type X in healthy versus disease states. So here are our example with respect to COVID-19. So these are some of the work that I have contributed in the context of COVID-19 since the beginning of the pandemic. So we were able to identify potential tropism of the viruses in like potential cell types. So we identify epithelial cell in the nose, for example, as a potential site for the virus tropism at the beginning of the pandemic, and later on, once we got our hands in the infected sample, we were able to do systemic profiling of immune cells cross-sectionally, for example, looking at the immune cell across different severity, from symptomatic people to mild disease to severe disease. We were able to look into the local and systemic immune response in adults versus in children, and what could contribute to, for example, more asymptomatic or milder disease in children when compared to adults. We also profile immune cells or immune landscape in the model of human SARS-CoV-2 challenge, which is under review as well. So these are some of the application of how single-cell RNA sequencing could be applied as a technique to really understand immune responses in the context of the disease, and ending up in a relatively impactful findings here. And then with SARS-CoV-2, there's a lot of creative ways of how we would be able to apply the data, just existing data, right? To look into more things that we would be able to. So for example, we know that SARS-CoV-2 is a virus, it's intracellular, it can result in the expression of viral RNA within our single cell, right? So in addition to us trying to map the reads to a human reference, so what's about we try to map the reads to viral references as well? And then it was successful, at least in case of SARS-CoV-2, where people were able to identify SARS-CoV-2 RNA in the single-cell RNA sequencing data. So now we can ask more questions, right? Like what what cells are actually virus infecting, and what would be the changes or differences in phenotype of the cell that are infected by the virus versus the cell that are not infected, right? So we could ask a lot more questions based on this creative way of analyzing the existing data, and we actually doing this more in our own work as well, because we right now we're doing a lot of single-cell RNA sequencing in the context of infectious diseases. So not everything as easy as detecting SARS-CoV-2 RNA. There are a lot of consideration to think about, for example, whether a viral RNA would have a poly-A tail or not. So a lot more things need to be optimized in order to do this kind of analysis, but it at least demonstrates the potential, right? Like additional information that is already collected. We just need to be creative in terms of how we use the existing data. We also have, in addition to the context of SARS-CoV-2, we also are doing a series of experiments in the context of dengue virus infection. So this is a paper that was published in 2022 based on preliminary samples, but we were able to identify longitudinal changes during acute infection. And what we were able to show was that over a day, right, there are dynamic changes in immune response, and that could contribute to the severity of the disease. So lots of things could change over a day. So now, beyond transcriptomes, can we detect other omics at a single cell resolution? And as a Ponpan, as a Ning, also touch a little bit about like other things that we can do. So one other thing that might be of interest for immunologists would be sequencing the VDJ receptor. So basically the TCR and BCR sequences, right? So to illustrate, right? So we could think about TCR or BCR sequences as natural biosensor of health and disease, right? So once we get infected or we get immunization, right, like our repertoire of T or B cells would change, and then we have T or B cell that would be specific to a given antigen that we receive. So when we think about the TCR, BCR, there are a couple of different ways that we could think about. We could think about binding to a specific antigen, right? So this is based on a given sequence and linking that to a specific epitope based on an upcoming pathogen or an upcoming vaccination, for example. Or we could think about the sequence, so not based on a individual sequence, but based on the whole repertoire. So we can have a repertoire from like healthy people versus diseased people, and then the profile of repertoire could tell us about, like whether we have any signature of any disease, and is there anything that could be intervened therapeutically. So there's been some companies. I'm showing you an example of a company that trying to decode. This one is based on the TCR sequences and try to, based on a lot of information that we started to collect, especially during the pandemic. Lots of SARS-CoV-2 related sequences have been generated. Right. So, like now we have machine learning, AI models, so a lot of people trying to make use of this rich information to see whether we would be able to infer any diagnostic tool or therapeutic tools based on information that we have. And then as a Ponpan would mention later on about how we are trying to do this immune phenotyping and immune repertoire sequencing in the Thai population. And then the additional information we could get, for example, we could get a single cell attack-seq. So this one is to profile single cell chromosome susceptibility. So the basic idea of droplet-based sequencing is similar, but instead of looking at RNA expression, we take a look at chromosome accessibility instead. There were protocols that we can do single cell chromosome accessibility and the single cell gene expression profiling within the same cell. So we would have the multi-omic information based on different layers from the same sample. And then we can also do what's called protein expression within the same cell. And the idea is to some extent similar to traditional method where we use antibodies, right? But when we think about antibody, we use for flow cytometry, we tag antibody with fluorescent dye. But here we just tag specific antibodies with DNA, right? So this is like a barcode. So if we have cell that bound to specific antibodies, then we would have the DNA barcode, right, being included in the droplet. And then once we perform the sequencing, we know whether there's a presence of that particular antibody or not based on that barcodes that we have when we tag the antibodies there. So beyond single-cell RNA sequencing, right, there's some more and more interest in another whole new area, which is the spatial transcriptomics, where not only we are going to get transcriptome from cell, but then we are going to get information about transcriptome at specific location. So right now, there are typically two major approaches. So the first approach would be sequencing based, where we have probes or tag that has barcodes based on specific location. So once we capture the cell, then the sequences that are based on the location would be incorporated into the transcriptome profile that we're going to get. So this sequencing-based approach has some like pros. That's because we sequence the RNA there, so we could still detect the variability, the allelic differences in there, and we would be able to detect the whole transcriptome in there. But this is at the expense of lower spatial resolution. So for example, the technology that I'm showing you here, like 10x VCM, so each spot that you're seeing has the range of 1 to 10 cells. So like not really single cell yet, but there's another area of transcriptomic approach, which is a fish-based, so just imaging-based, right? And this fish-based is based on probe, right? RNA probe. So you need to have some idea before to decide probe specific to genes of interest, right? So like whether you get whole transcriptome or not, it's based on the design. It requires a lot of optimization. But the pros is that the resolution is really, really high. So now the resolution is at the level of subcellular compartment. So now is another hot area, and it's being applied a lot in the context of cancer biology and cancer immunology. And there's some labs in Thailand who start doing this as well, and we've been involved in analyzing the data. And as you can expect, right, like having information on single-cell RNA-seq and spatial omics, they can be integrated together, providing even more powerful analysis pipeline that would even enhance the discovery of anything new in this particular experiment. So this example of the sequencing-based spatial transcriptomics, where we have a slide, and then the slide would have kind of DNA probes that or DNA like hair like this, right? Just like like in the 10x chromium, where we have DNA on the beads, but here we have the DNA on the slide, and then we have a poly DT that poly DT that would capture poly A tail, and at the same time we would have a barcode specific to the location. And this is what we are seeing in the result. And then we could also do, because this is transcriptome, do similar things, just like what we did with single-cell RNA sequencing, doing the cell type clustering, cell type annotation, and then map back to the location, and then identify, like which cell type is where, and then would there be any differences based on different areas in the location of the given tissue. And this is result from the fish-based approach. As I mentioned, right, resolution is much higher. You can see expression of particular RNA within specific location of the cell. You can do things called cell segmentation, modeling on the boundary of the cells, and then see where a given RNA are within the subcellular location of the cell. So some of these are the technology that are like highly competitive at the moment, but at the same time, it could provide us a lot more, right? So not just transcriptome or the cell, and then the location, and then the resolution is really high. So this is an exciting area. And in fact, the methods that we just mentioned, right? Single-cell sequencing, single-cell multiomics, single-cell spatial transcriptomics have been named Method of the Years by Nature Method in 2013, 2019, and 2020, respectively. So now I'm going to hand back to Ponpan to talk about our applications in Thailand.
Okay. So now that you've learned quite in depth on what can the single cell data tell us. Here comes some application. So with this advancement in technology, there's a group of scientists formed together a human cell atlas, which is quite similar to Human Genome Project about 10, 20 years ago. So now scientists come together and use single cell technology to try to map every cells in every tissue of the human. So the aim of the Human Cell Atlas is to comprehensively perform reference map of all cell type in all human tissue. And there are several bionetworks of the Human Cell Atlas, or HCA, and one very active one, and probably the first one, is the Immune Cell Atlas, where we try to atlasing all the human immune cells systemically and also at various locations in healthy and in disease. How about in Asia? Actually, Asia is home to most of the world population and a very diverse population within Asia, not just one Asia, but we have the Chinese, Indian, Thai, which has so much difference in genetics, ethnic and cultural, environmental background. So within Asia, we have this Asia diversity, trying to address the gap that Asian population genetics data is underrepresented in the world genetic data. So as you can see here, Asians account for only 1% of genetics data since like Human Genome Project, while Asian population are the majority of the human population. So in trying to address this gap, we form a network called Human Cell Atlas Asia. And the first project that we're doing together is the Asian Immune Diversity Atlas, where we aim to build an atlas of Asian immune cell types across various ethnic background in various countries. And these data will complement with disease cohort that's relevant to each local communities as well. So in this project, first, initially, Singapore, Korea, and Japan formed together in the first phase. And then we joined them, Thailand and India joined them in the next phase. And now current phase, we have many more countries joining. So within Thailand, our first phase, we have samples collected and done at Mahidol University in Bangkok. But in the second phase, we expand collaboration and also disseminate knowledge into other part of Thailand as well, with a lot of very nice collaborators here. So what is the impact? Why do we have to do this? So for having this IDA, we are able to technology transfer from other Asian country to Thailand, and we are able to address diverse genetic background. And importantly, we have unique healthy Thai database that will be available, and that will be important for later on compared to disease and later on development of biologics, biosimilar vaccine, and all the diagnostics. That's quite diverse between population. So this is just to let you know that we're still recruiting new donors. If you're interested, just let us know. So this is the recap now that we know about single cell omics in immunology. Actually, we not only healthy, but in disease context that as Waradon mentioned, that we can apply single cell profiling in discovering disease molecular mechanisms, identifying drug targets, develop diagnostic, prognostic biomarkers, identify drug resistance or responsiveness in precision medicine, assess probability of efficacy in candidate biologics, and also with immune repertoire, we can also aid vaccine design and therapeutics such as monoclonal antibodies or CAR T cell design and intervention. So these diseases involve that in immune response, such as infection, inflammatory disorders such as autoimmune disease, aging, transplant, and cancer. So I'll end this with our own journey of single cell. We started in 2017. Well, we start collaboration with Sarah Teichmann, who is the leader of the Human Cell Atlas and actually the pioneer in single cell genomics in the world, actually, at Sanger Institute. So we established single cell lab. We have some workshop to train other Thai scientists who are interested. And then we become a part of HCA Asia and finally hosted HCA Asia annual meeting last year. Through this, we got thanks to several funders and institutes, especially the Newton Funds and also Thai funders such as PMUC and STDA, who gave us support for establishing the single cell omics and also the work in dengue infection and also now expanding to other areas as well. And for IDA, we also got fund from CZI, Chan Zuckerberg Initiative. So now we have our facility for quite some time. So we're acquiring the high throughput droplet-based single cell. And also we have tried other platforms as well, such as the SmartSeq2 I mentioned earlier. Thanks to all the funders. I probably don't need to mention each name at a time. So our group is expanding right now. We have 4 PIs and a lot of wonderful research assistants and graduate students and postdocs. I can't be thankful enough for the great team we have here.
With this, I'd like to thank.
Oh, sorry. So, and also would like to thank all the collaborators, which I mentioned along the way. And if you want to learn more, we also have a graduate programs. If you're interested, you can give me a call, contact me or email me. Okay. So I think that's all we have. We spare some time for question session.
Wow, thank you so much, Ka, for giving us very comprehensive background and also share with us with the recent, technology. And actually, our team consult Janine to help us with the single cell with the umbilical cord sample. So yeah, I would like to tell everyone that if you need some help with the technology, I think you can consult them. So we personally got that experience. So thanks very much. I would like to open for any question from the audience. Are there any question from the audience? Maybe I can start because, okay, I can see one open their camera.
Yes. Hi, I am a Ph.D. student from Sira Hospital, and I have one question about the sampling of the sample size. Why we want to design the single-cell RNA sequencing in the time of the cancer study.
So the question is how many
How
samples?
many sample size is enough for the studying about mechanism of action in the particular cancer? Because I'm right now I'm studying the pancreatic cancer to understand the intratumoral heterogeneity in the patient. So if I want to design some kind of experiment, how can I consider for that?
So it really depends on the. So you mentioned the heterogeneity, right? So it depends on the question that you're asking, right? So are you just going to take a look at the atlas in your very specific context, or you want to do a comparison? And if you want to do a comparison, then how many conditions, right? Different conditions you're looking at. So, I could just mention, like for, when I was a postdoc in the UK doing the COVID-19 study, like we could do like over like 100 like individuals, right? Like it was super, super expensive experiment, but at the same time, because we have so many samples, then we can be lenient about, like the variability. We just collect, like the metadata, and then we can adjust things statistically. But in the dengue cohort, thanks to Ajahn Ponpan, so there were only 24 samples and then 3 severity groups. So in that case, like things had to be very, very well curated. So viremia was similar, the timing of the disease was similar. So we tried to control all the confounding factors. So now that was a limitation, right? Like on the samples that we can collect. So fortunately, so as Ponpan mentioned as well, that now we can multiplex sample. So in the past, 1 sample, 1 length of the 10x, but now we can pull more. So I think that like 8 to 12 samples right, per each length. So you can expect that typically we pull cells and then we run about 10,000 cell per length. So you can expect that, like if you have like sample from, I don't know, like 10 different sample pulling into 1 length, you get like 1,000 cell per sample. Would that be enough to capture the heterogeneity of your sample, right? So that depends on the existing heterogeneity that you expect to see. So for example, PBMC, like 1,000 cell per like sample, that's enough, right? But then, like if your cancer cell are heterogeneous and you want to see like really rare cell, right? Like the cells that you're really interested are really rare, then you might need to do, like the higher multiplexing, or, like more sample being multiplexed together.
Yeah. And higher number of cells per sample as well, in order to see like rare population in each sample. So it's all, we probably can't tell you how many sample is needed, but it's also because it also depends on your research question and your data, what you want to compare, and how similar or how different each groups are. So if the difference is not much, you probably need more N to increase the statistical power. But if your difference is quite clear, you might not need that many. And if each one is quite homogeneous, each group is quite homogeneous, not that much variability, maybe you don't need that many in order to see the differences.
Yeah. So let's say like, like your context is cancer cell, right? And if you just want to look at cancer cell and you collect your isolate cell from the cancer themselves, then you might not need that many samples. But if you also interested, if you have hypothesis on rare endothelial cell in the cancer, or super rare stromal cell, then you might need to collect like more cells, right? So you need to have like, like much bigger scale of the sample collection.
Or if you want to look at responder and non-responder to treatment.
Thank you so much. Thank you so much for your advice.
Okay. Oh, by the way, I should mention that besides the IDA project, we are now expanding to also collect tissue samples so that we can have reference of tissue, and we'll probably start with colon. And there's also a consortium within HCA Asia that work on Asian Cancer Atlas. So if there's anybody interested in contributing on samples, or maybe also on like single cell work, please don't hesitate to contact me, so I can like help liaise with the consortium.
Yeah, can I ask, like, to collect tissue, how do you collect it? Does it has to be fresh, freshly processed or something?
All right, that's a very good question. It also depends on what we want to proceed next. So the best one would be fresh tissue. For cancer, it would be a bit more difficult than PBMCs because it's in tissue and we need to dissociate them. And protocol in dissociation actually affect the results, the transcriptomes results. So when we're doing these atlas or profiling the tissue across several countries, the protocol need to be standardized. So so the fresh part would be good for like RNA sequencing from cells. But if you have frozen tissues, one thing we can do is single nuclei RNA sequencing or ATAC. So instead of isolating cells, we can isolate nucleus, which is more suitable for previously frozen tissues. Right? Correct me if I'm add on if you want to add anything.
Yeah, so typically FFPE tissue. So in the past, like some of the spatial omics could handle that quite well. So that's why people in the cancer field like having lots of spatial FFPE tissue, like they tend to do like spatial approach instead of like single cell approach. But the field is moving as well. So there was just a recently like advertised kit from 10x claiming that they could do process single cell RNA sequencing data better with even like FFPE tissue. So we haven't tried that one yet. But like it's there and the field is moving, and you can expect that the quality or the information that we would get right from the FFPE or even fresh frozen might be inferior compared to processing from just fresh single cell. But then like if the context like limit that, then it's also possible to process the sample that are like frozen or even FFPE.
Even FFPE is right for spatial too. Yeah.
That's very interesting. Are there any other questions from the audience? So when we do fresh cell, that means we run each sample at different times. There are no problem with the analysis, right?
So actually we've been through this several ways. So so if we run each samples at different days, it's also subject to batch effect between each runs, right? And there's actually so first of all, we need to standardize the wet lab protocol, and also in analysis process, they have batch correction steps that can try to batch correct. And also we've tried this with some cancer tissue with freshly prepared and single cell. That would be best for the quality of the cells, but it's not always possible logistically. When we get the sample, it might be in the afternoon. It takes time to process, digest, and then prepare cells into the machine. So it's also possible that we dissociate the tissue right away and then keep it frozen in dissociated like single cell, and then keep it frozen as single cell, and then thaw it and perform experiment the next day. So and they're also possible that they have bank of frozen tissues with various protocols, and then we thaw out the frozen tissue and try to dissociate them, and then subject to 10x. And lastly, FFPE tissue. So that's so many protocols that we can do with differences in the quality of the data. But sometimes if you have bank, it might be more important to be able to use a lot of collected samples that can provide a good enough quality data than to try to do just fully fresh, but you don't get that many samples, so you just have to wait.
Yeah, so as Ponpan mentioned, right? So at one point, it requires like very well controlled experimental design, but then in reality the limitation. But there are computational tools to adjust that. But to be sure that computational tools are handling this correction well or not, it depends on having a good benchmark data set, right? So now we're getting more and more like reference level, right? So I mentioned tabula sapiens, right? Trying to collect tissue from the same donor, like minimizing this like batch effect. So once we get like reference, like this many cell type in the blood, this many cell type in the lung, right? So we could use that reference as an anchor, and then even if we have query data that we just generated, we map that back to the reference and use that reference to kind of like adjust the lower quality data. So I think like that's the idea, that like once we get the good like gold standard reference, then even if in the future we have like substandard data, then we could use the gold standard reference to kind of correct that. So that's why, right, collecting more and more and being like super serious about generating references in the IDA, where I think that they really control for the experimental standards. So that would really help future experiment where we don't have to worry that much about like the quality, as long as we map back to the standard.
Okay. I think single cells are quite new to us as a community, as you say. So probably we're going to learn more. Oh, I see another camera open. Dr. Tawisak.
Thank you, ครับอาจารย์ It's a very fruitful talk ครับอาจารย์ It's quite clear. So actually we have some project that we collaborate with อาจารย์ยอด and อาจารย์ชนิตรา and we face the problem when we use the cells to do the single cells analysis. I think you already know the problem is we try to compare the condition, but it seems that the cell is not enough. I mean, one condition is have a high depth of sequencing, but another one is maybe like a very, very low depth. So it cannot compare it. So how we can solve this problem? This is the first question. So do you have any suggestion about that one?
So probably we don't know enough about the experimental conditions. I should mention that Ajahn Yod and us are the same, and we kind of together in most of the project. So please feel free. Maybe we need to know more detail on the sample preparation steps, as well as different QCs level. So please feel free to contact me or Ajahn Bloom or through Ajahn Yod, or you can set a meeting and then we can help troubleshoot.
Yeah, sure. So another question that in term to do the analysis, I'm not sure I read somewhere and I can't remember about the reduction of the dimension when we do the analysis. So I think it have some, they just raise some problem that if we can we trust that one or not, or as you mentioned about the reference, that it get more data is can lead to more reliable to reduction of the dimension, right?
Yeah, so I think there's some some scientists who think some kind of dimensional reduction tools are kind of so random, but at the same time, so I think we kind of more toward the middle ground, that we have to be careful with our interpretation, because in the end, right, we see cluster, and then when we take a look at gene being expressed in the cluster, then we know that this is a cell. We can convince ourselves that this is actually the cell. So it's not that it's totally random. It generates some usefulness that we can trust ourselves that they are real. Are there going to be better methods in the future? Maybe, and we hope to see that. So I think Ponpan and I we joined the field from the beginning, and the method we used 5 years ago. Now we know that it's a crap, but then we still infer some good signal from there, and then some of the strongest signal, right? Even the methods are not optimal, but then the signal are strong enough that we can extract something. But now, right, with the newer method, of course, things are going to be more accurate. We can distinguish, signal and noises better. So we hope maybe the next 2 years, 5 years, right, they're going to be better and better too. But then we can't take things that we need perfect approach to analyze the data. We use what space at the moment. and then acknowledge the limitation. And I think maybe that's enough for, like the progression of the field, and hoping that, like the next, so even now, right, we're not just tools that are available today. Like we still, exploring new tools that keep coming out every single day.
Yeah,
So-
the field is still advancing quite rapidly. And
Yeah.
yeah. So when we get the data, actually the data has to pass several steps of QCs. So such as like we cut out high percent mitochondrial genes to QC for dead cells, so remove dead cells, and also look at N genes per cells and things like that in order to like make sure that our data quality is good. And then there are also other steps such as removal of contaminating soup RNA in the ambient RNA and things like that. So when you do UMAP, dimensional reduction, and clustering the cells, there are also several ways that Ajahn Waradon mentioned. So both unbiased way and also the reference-based way. So we actually use both methods depending on our research question and sample as well. So in PBCs, we can freely use the reference-based method, unless if you have like certain cell types that's specific to your condition, because the reference for PBMC is so good. But if you work on special type of tissues, maybe it's better to use an unbiased way so that you can discover unique cell types in the sample.
So so nowadays they just have like a database of the PBMC from the normal tissue, right? Normal people, right? But not in the cancer patients, right?
Yeah. So I think the at least the PBMC, the gold standard database. So like the reference is as good as the reference, like the data set, right? So I think the PBMC, and I think the airway, the lung, I think people believe that they're the gold standard now because of the quality of the references. It's really, really good. But you're right. It's just based on the healthy. So how we're going to apply to the disease context, it depends on like take a look, right, and see whether things make sense or not. So we are having some analysis ways of take a look at immune infiltrates in like all the tissue. Can we use friends from the PBMC, but then we have like granulocytes, or how we're dealing with it. So it's an ongoing process as well, right? How we're going to incorporate like unsupervised versus supervised doing things together. There are like new framework, but then we also try like our own framework as well. So it's moving, and I don't think there's a correct answer, and sometimes the good solution for one data set might not work in a different context. So even for our people, our students, like like they try like multiple tools and then just look, right? So there's no way that you have to do this 1, 2, 3, 4, and then sometimes with this like batch correction, they try like 5 different methods and see like how things look like in the beginning, and things could be different from the different data sets.
Okay. So thank you. So the last one, so nowadays our lab just like do the project that we collect the liver cancer cells tissue. Actually, we just collect the normal one and then the cancerous one, but actually the goal we want to like develop the cell line and also develop the organoids. So if like we can move on to the single cell, is very interesting because nowadays we just keep the samples like for do the whole genome sequencing. So I think that process is not suitable for the single cell approach, right? So if we like want to move on, maybe we have some collaboration later. Thank you very much. It's a very nice lecture.
Collaboration. And I should mention that as a community of scientists, we're trying to build this atlas. Now we started with healthy, but now we started with healthy, and we have quite a few healthy already, and now the few is moving towards disease. So maybe in the next 3 to 5 years, we will have cancer at last for every organ, and then you can compare particular cancer from different ethnic groups and things like that. So we all could, like, as a global community, we could all contribute to the data generation.
Okay. Well, thank you so much ค่ะอาจารย์หนิง and อาจารย์ปลื้ม it's a moving field. We learn new things every day, and I hope that this talk will initiate some interest from our team and anyone who would like to get more information and collaborate with them, please feel free to contact them directly. We will keep this record in our website, and I will be very happy to link with them if anyone needs their help. Thank you very much, Ajahn Ning, Ajahn Bloom, and hopefully we can invite you back again, maybe with the Atlas of Cancer sometimes. Okay? Okay, ค่ะ. Thanks everyone for joining ค่ะ